Are you ready, Scott? Cool. A lot of people tonight.
Oh, no,
Oh, he told me he was coming.
just in time.
He told me he was coming. Were you at Gabe's?
No, I was gonna do that, but I fell asleep. Those are some fun pants.
All right, let's get started. Topic 5. So exciting. All right. Do it. Okay. Okay.
Okay. So topic 5 is cell signaling. So this has to do with a lot of signals that are coming from outside the cell, and usually the types of signals that we're focusing on are hydrophilic ones that can't cross the cell membrane, which is why we're dealing with a lot of like cell different types of receptors and the signaling pathways that they activate. You can have internal signals, but we don't really go into those as much. Some of the different ways that we can regulate cell signaling pathways is through crosstalk, which is when you have molecules from other pathways interacting with the pathway that you're focused on. So it's like two separate pathways interacting with each other, and that's how you can, maybe enhance a pathway or shut it down. There's also amplification, which is usually when a cell signaling pathway has multiple steps and you're able to get a broader response based on the single signal that is binding. So you can have different outcomes. I already mentioned amplification. There's also phosphorylation, which is phosphorylating different molecules. And they can cause conformational shifts in the molecules that they are either, well, when your signal binds a molecule, that will cause a conformation shift. And then all of these subsequent signaling steps in the pathway can cause conformational shifts in proteins and enzymes changing their function. So two of the primary functions of cell signaling are to change protein function, as I just said, and then change protein levels in the cell, which would be through modifying gene expression. So the first pathway that we talk about, and feel free to stop me at any point and ask any questions, is the RTK RAS MAPK pathway. So these involve kinases, and I'm just going to quickly review kinases in case you guys aren't very familiar. They cleave off the phosphate group from an ATP and attach it to another substrate. This is typically onto the hydroxyl group of an amino acid, which would be like serine, threonine, or tyrosine. And that's basically all you need to know about kinases. If I'm going too fast, also feel free to slow me down at any point. Yeah, absolutely. Sure. I don't think she would ever ask you, like which amino acid is getting phosphorylated, but sometimes it's helpful, especially since we'll be talking about tyrosine kinases. That's something a tyrosine is obviously getting phosphorylated with those.
I know it happens on the hydroxyl group. That's like very important. Please don't mention that it's something ionic bond formation to phosphorylate because that is wrong and she'll pick us off.
Yeah, basically, I mean, I don't think she would ask you like, oh, which group does it attach? Yeah. Oh, she does. All right, never mind. Never mind. But yeah. Okay, yeah. So yeah, know that it attaches to the hydroxyl group of an amino acid. Those are the three that have hydroxyl groups on them. And all of the slides and everything will be posted after as well. So getting into the RTK RAS MAPK pathway. So this is obviously a cell signaling pathway. It's very important. This is one example where you have amplification that occurs due to the multiple steps that are within the pathway. And the specific function that it has for the cells is for cell division or differentiation. This will cause changes in your protein activity based on the proteins that are being phosphorylated. And will ultimately end up in changes in gene expression because again, this will either cause your cell to get ready for cell division or cause the cell to specialize and or differentiate. I'll go back. This one. And I find these images really helpful If there's something that you would want to draw out on your cheat sheet for exams, I think that's really helpful. Or if you're not as much of a visual learner and you like writing out the steps, that's one thing that I would always do on my cheat sheets. Instead of drawing the receptors, I would just write out the steps for each signaling pathway, which was just more helpful for me. But if you're a visual person, these diagrams are also really helpful. So this pathway involves RAS signaling. So the first thing that happens here is that you have your signal molecule that binds to the RTK receptors and it causes them to dimerize. So there are two separate molecules that join together upon signal binding. The dimerization allows for cross-phosphorylation to occur. So these are on the tyrosines of each dimer. They're able to phosphorylate each other. And then once this phosphorylation occurs, you have your GRAB protein, which is an adapter protein. So it doesn't have like any catalytic function in this pathway, but the GRAB protein has what's known as an SH2 domain, and that's a phosphotyrosine binding domain. So what would we think that a phosphotyrosine binding domain binds to? And specifically phosphorylated tyrosines, yeah. So the SH2 domain in GRAB2 allows it to bind to the phosphorylated tyrosines on the RTK intracellular receptor. Also, just a quick note, so in this diagram, you can see there's an extracellular portion, which is where the signal binds. And then you have it goes across the cell membrane and then has an intracellular portion as well. And so when the signal is being transduced, this is a question that Laura likes to ask a lot. Signal transduction does not refer to the actual signal molecule coming into the cell. It refers to the information that the signal is conveying and how that crosses over into the cell. In this case, through the activation of this cross-phosphorylation, which leads to the activation of the entire pathway. But yeah, that's just how the transduction occurs for the signal. So the GRAB2 protein has not only an SH2 domain, but it also has an SH3 domain, which allows the SOS protein to then bind. And this is the RAS activating protein. So the SOS has what's known as a PXXP motif. It's a polyproline helix. It's just a type of secondary structure. She wouldn't ever ask you to like identify it or describe it. But you need to know that the PXXP motif on the SOS protein is able to bind to the SH3 domain on the GRAB2 protein, which also has an SH2 domain that binds to phosphotyrosines. And again, all of these binding interactions occur between these molecules like in a sequential order, because there's phosphotyrosines, GRAB2 is able to bind, which then allows the PXXP motif of the SOS protein to bind to the SH3 domain. So this is important. Slow down. Okay. I'll wait. Sorry, guys.
So basically, guys, GRAB2 is just there. So that the end goal is for you to like receive that signal and pass it on to RAS. GRAB2 is just there because SOS doesn't have an SH2 domain. And so that signal from the signal molecule would not be able to get passed through the RTKs to SOS because there's no way for the SOS to find that phosphotyrosine.
Yeah. And the thing that's important about bringing the SOS to this area is because it's just freely diffusing around in the cytosol. It's not transmembrane protein. It's not in any way anchored to the membrane the way your RAS protein is. So it's important to have this GRAB2 protein bind because then it localizes your SOS protein to this area, bringing it in closer contact with your RAS protein so that it can activate RAS. Does that make sense? Does anyone have any questions so far? Okay. So like that last bullet says, we're bringing SOS to the membrane so that it's in closer contact with your RAS protein. So the specific, what's going on? Oh, okay, there we go. The specific activity of SOS is that it acts as a GEF, which is hopefully something that you're familiar with from the nuclear import and export, which we talked about. So a GEF is a guanine nucleotide exchange factor, and what that does for RAS as well as RAN. RAS and RAN are very similar to each other. They're not the same, but they are very similar. They both are GTPases, which means they hydrolyze GTP to GDP. And as a GEF, the SOS protein helps remove GDP from the binding site of your RAS so that GTP can diffuse into the binding site. So again, just to kind of like keep reiterating everything, because GRAB2 was able to bind to those phosphotyrosines, you're bringing SOS, which is your RAS activating protein, to the cell membrane where it can cause GDP to leave the binding site of RAS and GTP can enter, thereby activating RAS. Is that good for everybody? Okay. So now that RAS is active, it can go on and go through this signaling cascade, or at least continue the signaling cascade. So the activated RAS is when it's bound to GTP, and this causes a MAP kinase cascade. And this is an example of like the signal amplification that we mentioned earlier. MAP stands for mitogen activated protein. It's a type of growth factor. That's why this pathway is involved a lot with cell division.
Later on, we get to DNA, cell division and stuff. We're going to go over a couple more proteins that are widely studied in cancer genetics. But like, I don't want to get too far into it, but there are proteins that are present in pathways like these that serve as further on genes and proliferation like crazy. Just keep in mind, they're important. Don't forget. Yeah.
So yeah, just know that this pathway is important for cell division. And if you were to have some sort of mutation in this pathway where you can't shut it off, you have a gain of function mutation. And if it leads to cell division, that might lead to a cancerous phenotype. But that'll come into play, I think, later on when we're going over cell division, but it's a good thing to start thinking about now for sure. So the MAPK cascade, it involves 3 different kinases. We start with MAP kinase, or MAPK3.
Don't call it MAPK3.
Yeah, don't, yeah. I think that goes without saying, but it's on the recording now. All right, so the activated RAS, which is GTP bound, will activate the MAPK3. This is the only mechanism that is really still not very well known. RAS is not a kinase, just so that's clear. RAS does not phosphorylate MAPK3. They don't really know the mechanism by which RAS is able to activate MAPK3. We just know that it does. MAPK3 is a kinase, remember, MAP kinase. And the role of a kinase is, again, to phosphorylate molecules. So MAPK3 phosphorylates MAPK2, which activates it by causing a conformational shift in the protein. MAPK2 then phosphorylates MAPK, and MAPK can then go and phosphorylate its downstream targets, which, if we're thinking about how this would lead to cell division, it would probably be transcription factors that are going to specifically cause cell division. Yes?
Well, I just know in the first slide you said pathway, MAP2A.
It was just a typo.
Oh yeah, that's, yeah. It should say MAPK2. Yeah. Yeah. Yeah, sorry about that. We'll fix that in the slides. Any other questions? Okay. So this cascade allows the activation of usually transcription factors leading to changes in gene expression, which could lead to cell division or cell differentiation. And there is a RAS gap. It's not shown in this figure, but there is a RAS gap that can help shut down this pathway by activating the intrinsic catalytic ability of RAS, which would be to hydrolyze the GTP to GDP. So again, this is very similar to RAN. Hopefully this is still somewhat fresh from exam 1, but a GAP is a GTPase activating protein. Again, activating that intrinsic GTPase activity. Yes.
Makeshift soup.
Yes.
How does that conceptually happen, Sky? I don't understand.
How does GAP?
How does RAS-GAP being present stop the pathway?
Because RAS-GAP, thank you for your question. RAS-GAP again stands for GTPase activating protein. RAS doesn't technically need a gap to perform the hydrolysis of GTP to GDP, but this can help speed along this process. It activates that catalytic ability. The gap is not itself hydrolyzing the GTP, but it will bind to your RAS protein and then cause the catalysis to happen. And then you have GTP forming into GDP, and now GDP is the inactive state of RAS. When RAS is bound to GDP, it is inactive. Does that answer your question? Okay, so this is just a summary slide. So just to go over it very quickly, and then if you guys have any questions about it, again, we have the extracellular domain of the receptor, which is where the signal binds. It causes dimerization. This is really important to know this specific order. You have signal binding causing dimerization, and then the dimers are close to each other, they've dimerized, they're able to cross-phosphorylate each other. So if signal is not able to bind, you won't have cross-phosphorylation. If you have somehow a mutation in these tyrosines, signal can still bind, it can still dimerize, but you can't cross-phosphorylate. So knocking out the ability of the receptor to cross-phosphorylate is not going to affect signal being able to bind. I think one of the most important things that Laura really tries to hone in on in exam questions and problem set questions as well is making sure that you know the order that everything is happening in. So after cross-phosphorylation, you have the GRB2 protein binding, then SOS. SOS is able to act as a GEF, activate RAS. RAS can go activate the MAP cascade. So again, know the order in which the pathway occurs, write it out, know where each molecule is. RAS is anchored in the cell membrane, whereas SOS is floating around freely in the cytosol. Yeah, does anyone have any questions on this pathway? No, we're good?
I remember in the video, people like using different names and like kind of got confused.
Okay.
The same thing.
Cross and auto phosphorylation.
Yeah, there's like zero phosphorylation or something like that, and like I was just kept on like switching between those words.
Yeah, so cross-phosphorylation, autophosphorylation, it all refers to the same process of those, of that tyrosine kinase ability. Was that specifically it, or was there something else? Okay, yeah. Okay, so we'll move on for now, and then we can come back to that. Okay.
All right. When you get the questions, just let me know and I'll get her back up. Okay, cell survival signaling pathway, my favorite pathway. I don't know, I did like a little project for Laura on one of the proteins present, so I kind of like it. So why do we need the cell survival signaling pathway? I don't know, life's kind of sad, and the default for all the cells is programmed cell death. So this pathway specifically inhibits apoptosis, not necessarily like the process itself, but the triggering of apoptosis, and we're going to look at that in a little bit. And it's just a very, to me, interesting pathway. I had no idea this happened before Laura's class, and then kind of blew my mind. And then it's very important for the reason in class that we're focusing on, occurrence of crosstalk with other pathways. If your cell is triggering like cell death, then all the other processes in the cell also need to cease, not only because you're not trying to waste like materials from the environment, but also because just be useless. And you need to do it in a controlled way so that nothing wrong happens, and then
all of a sudden you have cell proliferation from a cell that's supposed to not be there anymore, which is how a lot of tumors happen. Okay, so here is the initial part of it. So beginning, survival signal binds to RTK. RTK dimerization and phosphorylation, same process that Sky went over just a couple slides ago. And then you're going to have this PI3 kinase binding via another SH2 domain, which Sky also went over. So you need a phosphotyrosine domain over there, and then you're able to bind it. And so the function of this kinase is to phosphorylate this protein called inositol phospholipid. Actually, it is not a protein then, but so that happens to be oh, you got it?
Yeah, actually it wasn't, if we phosphorylated, yeah, I think we are the same thing. I think,
Yeah, that's just referring to PIP3 having 3 phosphates. And so it's like the inositol phospholipid before the third phosphate gets put there. And then once the kinase phosphorylates it, it's just called PIP3. Acetyl? Well, it's either like inositol phospholipid, which is the protein pre-PIP3. Do you recall anything with acetyl in this pathway? It might just be activated inositol phospholipid, which just means PIP3. It's the same thing. So yeah. But so yeah, the purpose of this part is to get the activated inositol becomes PIP3. It's anchored in the membrane similarly to how RAS is on the previous pathway. And in the next step, we're going to see it becomes the binding site for protein kinase 1. And it's important for you to see the difference between the 2-phosphate molecule and the 3-phosphate molecule. So yeah. Question? No, the SH2 domain is found in the PI3 kinase, and the RTK is meeting the requirements for you to have that binding, which is having a phosphorylated tyrosine.
Remember, SH2 is a binding domain, so it's part of the protein that is binding to a phosphatyrosine. Okay.
Yeah. Anything else?
Oh, all right. So like, they don't, just because something like SH2 domain or like SH2 domain something, doesn't mean that the thing that it binds to that.
No, it just missed the criteria. So it meets the active site criteria for you to be able to bind that.
Same with like the PXXP motif, like RAB2 does not have a PXXP motif. It has an SH3 domain, which binds to a PXXP motif, which is found
In
on the
SOS.
SOS protein.
All right. So this step right here, you have the PIP3 molecule from the previous step. And so the PK1 kinase right there with the pH domain, which is what binds the triple phosphate, binds to the PIP3 molecule. And so the molecule that this kinase is trying to activate is also another molecule that binds to another PIP3. So you kind of need 2 of them for you to be able to like do this specific pathway. And so this PK1, which is the kinase, goes in, binds on its pH domain, and then there's going to be already an AKT present over there. And so this kinase right here is going to phosphorylate the AKT. It is very important for you to know this. The AKT doesn't get activated with just 1 phosphorylation. You need a second molecule or a second kinase to phosphorylate it. In this case, in this pathway, PK2, also known as mTOR, and then it goes on this side and phosphorylates AKT. And for that to be able to happen, the phosphorylation from PK1 has to happen first, and then that causes a conformational change on the AKT molecule, which then allows it to be phosphorylated by PK2 or mTOR, whatever you want to call it. Hi. That's the next step. Well, it dissociates once it's fully activated. Yeah. So yeah, PK1 binds the PIP3 molecule. AKT is already previously bound to a PIP3 molecule. PK1 activates it 1/2. mTOR comes in, activates it on the other side after the conformational change, and then it dissociates from the PIP3 molecule once it's fully activated. And then this is the apoptosis inhibition step. So the activated AKT double phosphorylated by the PK1, PK2. Dissociates from PIP3, enters the cytosol because previously it was attached by its lipid anchor. And so AKT comes in, binds BAD, which is this molecule right here, the one in blue, and phosphorylates it. And what that leads to is the dissociation between BAD and BCL2. What's important for you guys to know here? BAD is sequestering BCL2 like typically, so the normal is for that protein to not be activated by AKT or phosphorylated deactivated, my bad. And so what typically happens when you don't have this signal binding for survival, so once the cell reached dysfunction and then there's no benefit from having it anymore or just ran its course, that AKT won't ever be there. BAD is going to continue to sequester BCL2, and then BCL2 will not be able to inhibit apoptosis, and then the cell is going to start triggering its mechanisms for it to die. So here, the pathway we're studying, active AKT goes in, phosphorylates BAD. In this case, deactivating it with a phosphorylation, kind of like counterintuitive. Yeah, so intuitively we think of like, oh, phosphorylation means activation, not necessarily. And so it causes a conformational change. BAD and BCL2 dissociate. The inactivated BAD is going to be bound by another protein, 1433. I don't think, has she ever like asked a question about that? Yeah, I don't recall like anything specifically about that protein that I can think of, but it's important for you guys to remember BAD is then free. BAD is what's going to go in and start the process of inhibition of apoptosis, and that happens in the mitochondria, but that is a completely different process that we do not need to talk about because it's kind of complicated. If you want to look into it, the molecule being released is called cytochrome C, but you could do that on your own time. So BAD and BCL2, just quick again. Apoptosis, programmed cell death. BAD protein is the protein that allows for apoptosis because it sequesters BAD or BCL2. And then BCL2 is the protein that inhibits apoptosis whenever it's free from BAD. So there are two possible scenarios. Active BAD will be bound to BCL2, which then will inactivate BCL2. And then it'll go through the mitochondrial holes, and then that will lead to apoptosis eventually. And then inactive BAD, which is when it gets phosphorylated, counterintuitive, just write that down if you can't like.
Yeah,
like
don't always associate phosphorylation with activation. It just depends on what you're looking at. It can be either.
Yeah. So for this case specifically, the AKT is going to go in there and deactivate BAD through phosphorylation, which means that BAD and BCL2 are going to dissociate. Active BCL2 will then prevent apoptosis from happening. Any questions? No? Nobody? Okay. So then there's a little summary. I can run it through really quick. So the signaling begins when a survival signal binds the RTKs, and then it causes dimerization of the molecules and autophosphorylation. PI3 uses SH2 domain to bind the RTK, and then activates PI3 kinase. PI3 kinase phosphorylates PIP2 molecule into a PIP3 molecule with the third phosphate. PIP3 is stuck to the intracellular side because it has that anchor like RAS did on the previous pathway. And to be activated, it needs that third phosphate. And so PIP3 serves as a binding site for the kinase and then also for the AKT. So you need two of them. And so PK1, same thing, phosphorylates AKT, and then that causes a conformational shift. mTOR then comes in and phosphorylates the other side of the AKT or the other part of the AKT, whatever you want to call it. And then once both sides are, Both locations are phosphorylated. AKT dissociates from PIP3, and then it roams in the cell until its active site or in its active state. And then active AKT then phosphorylates BAD once it runs into it, which forces BAD to let go of BCL2 due to conformational changes, which change the affinity of the two molecules. And then BAD is then stuck to the 1433 protein, and BCL2 goes in and continues its cycle for prevention of apoptosis. Did I go too fast? Questions? You look confused, Lisa. The AKT and mTOR. I thought it dissociated after it's fully active. That would be a good question for tomorrow's lecture.
Yeah, so it says AKT is not yet active as a phosphorylation process. And to begin the keeping of the cell is
Okay, then we're gonna go with the dissociates pre-mTOR activation. Is in her lecture notes. But after that, it goes in, phosphorylates BAD, deactivates it, causes dissociation between BAD and BCL2. BCL2 goes in and inhibits apoptosis. So that's an overview of the pathway. I'd highly encourage you guys to either take a screenshot of Laura's slides, download Laura's slides, project them, get into like a group of friends and just run it through the pathways without any writing, without any listening. Just like go off of like what you see and how much you can explain. No. Draw it. You're allowed to draw it.
Yeah, you could put it on it. Yeah, but she doesn't give me so she gets
She still gives you the amino acid
yeah,
list.
it doesn't give you the it's not the pathway, she doesn't tell us.
Yeah, but I don't think you get the export and import cycles either. I think it's just the path or just the amino acids.
She's not gonna ask you about where I put, so that's why she's not. She'll give them to you again on the file, but she's not gonna ask you about it.
She might mention something around like, oh, RAS is related to RAN, and then explain like what happens in this part of the pathway and whatever would happen if this was shut off. She loves like if this would happen in the pathway, what would be the end result type of questions. So that's why also if you're able to like tell what each step does and what every molecule does in every step, then you're able to just block out whatever she's asking and like be able to say, oh, if AKT never dissociates from its PIP3 bind, then it will never be able to find BAD, and then BAD will forever hold on to BCL2, and then apoptosis is going to happen. Something like that.
Or like if she were to ask you like, oh, let's say you have both of these pathways that we just talked about and you added a phosphatase removes the phosphate in the RAS-MAPK pathway, you remove the phosphate group that will deactivate the signaling pathway and stop cell signaling from happening. But in this pathway, if you remove the phosphate, that's going to, rather than inactivate, activate BAD. So then, you just have to think about each subsequent step and how it's affected.
And she loves questions like those. So
yeah, in that case, BAD would be binding BCL2 and have it in the position of
So-
yeah, I mean, yeah, like
No.
overproliferative, like too much cell death would definitely be a phenotype of cancer that does occur, but it's not like it just not indefinite. It's just like a deleterious
process. Yeah, the more familiar you are with it, the more you can like, oh, that sounds wrong, like during the exam. Like you read something off of like the question and then you're like, oh, this goes this way. And then, but then this happens this way, explain why. And then you're able to tell it. So I'd recommend it. So new pathway, JAK-STAT, less cool than the cell survival signaling one, in my opinion. Kind of mid, but so that's the overview of the pathway. You know, whatever. So I'm gonna run through the steps. So beginning, it is involved in many processes, but before we go on, JAK receptors are not RTKs, okay? That is very important. They are not RTKs. They do not cross-phosphorylate each other. Janice. Shut up to my office hours earlier. So it is a receptor, lacks kinase domain. Write that down, engrave in your brain, tattoo it if you want. Not an RTK. Not an RTK. And so it has kinase subunits that associate with it. And those are called the JAKs in this pathway. All right, let me call someone out. Who's brave enough to answer a question? Yeah, all right, I'm calling Lisa out. What's a domain? Huh? That folds independently? Nice. Okay. It is also JAK right there, little blue thing, little blue guy. Also not an RTK. Let's not get that confused in the exam. Let's not lose precious points. Okay, engrave it. JAK is not an RTK. And the receptor is also not an RTK. Let's get both of those in there. The receptors in this pathway don't have any autophosphorylation mechanism, which is why the JAKs are present. JAK itself, just another kinase. That's literally its name. And JAK. And then again, sorry, I have to reiterate this. Not non-receptor tyrosine kinase. It does the catalytic activity for the receptors, and it is just a domain. Or it is, sorry, and it is a separate subunit that is not a domain of the original receptor. So sorry. Thank you. Oh, almost broke. All right. JAK1 and 2 are bound to either receptor. One on the left, two on the right. Prolactin signal comes in, and so the receptors dimerize. So with the binding of the signal, the receptors are going to dimerize, but how that differs from an RTK dimerization / cross-phosphorylation is they are not themselves able to cross-phosphorylate once the signal is bound and once they're dimerized. And so that is why the JAKs are important. Once the prolactin signal binds and they dimerize, the JAK becomes able to phosphorylate each other. And so once, oh Jesus, that is like so far down. But once the JAK protein is able to cross-phosphorylate, it then phosphorylates the receptor. And once the receptor has been phosphorylated, that is when the STAT protein is able to bind on either side. Can you show me the slide real quick? That's not. So, sorry about that. I just have to check it. Don't want to spread misinformation on the internet. So once that happens, the STAT's able to bind onto the receptor, and it also binds via an SH2 domain. What's an SH2 domain? Come on, it binds.
Don't want me to say it again.
This guy's gonna be upset.
No.
What's it bind to? Too late. You took too long. Okay, never mind. Okay. There you go. So what's it bind? With a phosphate group. So phosphorylated tyrosine. Okay. So once both stats are active on either side, they're able to form a dimer, dimerization process, and then that is caused by conformational change. And then they dissociate, well, before that, pre-dimerization, they dissociate from the receptor, and then they dimerize in the cytosol. And then they're able to enter the nucleus, and then continue on with their, what did Laura say? Whatever it is that they do. That is not a good way of saying it, but they are responsible for transcriptional regulation in the nucleus. And so they go into the nucleus. Do they need an import cycle? I don't think they need an import cycle. They just go in, right? Like the stats. I think they just go in. Yeah, I think they just go in. Don't quote me on it. We could probably ask her, but I think they just go in. So they don't need important to bring them. Actually, never mind, forget it. It is via important. I didn't check my own slides. But okay, so summary. JAK is a non-receptor tyrosine kinase, and the receptors themselves are also no RTKs. And they are located inside the cell. So the prolactin is able to bind onto the receptors dimerize. JAKs are then activated. They cross-phosphorylate each other, and then they phosphorylate the receptor. Once the receptor is phosphorylated, STAT's able to go in, bind through the SH2 domain that's present on the receptor because of the phosphorylation. And then once both STATs are active, they dissociate from the receptor, they dimerize, and then they go into the nucleus and continue on with whatever it is that they do, or more exactly, they regulate transcription. So yeah, this pathway is a little bit simpler than the two other pathways we were looking at. But she loves asking trick questions about dimerization, and please don't say that they are RTKs. Like, if you take anything from this presentation, they're not RTKs. I blame the slide recycling I did from previous years. So I am taking no accountability for that. It needs important. Forget what she said. All right. Oh yeah, and then for this specific case, prolactin has to do with milk genes. Milk, like milk, yeah, genes. So all right, there you have it. Any questions before we move on? Sorry. Anybody else? No, I'm kidding. Yeah. All right. Oh.
Last pathway. Okay. So the GPCRs, or G protein coupled receptors. These are just like the general things we're going to go over, specifically how in the GPCR pathway, we have things that act in a very similar manner to GEFs and GAPs. And obviously we're going to go over how the whole process works. So, oh, okay. All right, so the GPCRs are a 7 transmembrane receptor, and it's bound to a G protein that has 3 subunits, the alpha, beta, and gamma, the 3 most popular labels for everything in science. And the G alpha subunit, which is the most important one for all intents and purposes, is a GTPase. In the inactive state, the G alpha is bound to GDP because that makes sense. It's a GTPase. It needs to be bound to GTP to perform its function. When you have a ligand bind in the extracellular space to your GPCR, then it will cause a conformational shift. So it's a transmembrane protein again, so it causes a conformational shift on the intracellular side of the membrane. and this will cause the G alpha to let go of the GDP. Now GTP can diffuse into the binding site. Once the G alpha is active and bound to GTP, this causes it to dissociate from G beta gamma. And those kind of go off and do their own thing. They have other, sometimes in certain pathways, they serve a different function. Sometimes they don't really do much. We don't really focus on them too much. But the G alpha dissociates and can go on to bind to different effector molecules and cause signal transduction in the cell. One of the common examples of this is that G alpha will go and activate adenylyl cyclase, which is important in the pathway that makes CAMP, which is cyclic AMP. It's a very common second messenger molecule in the cell. But yeah, so now you have your active G alpha, it's bound to GTP, and deactivation of it occurs when you have RGS binding, and this will activate the intrinsic GTPase activity of G alpha, and it will hydrolyze GTP to GDP. Once G alpha is inactive, it will come back into this heterotrimeric complex with G beta and G gamma, and they will bind again to the GPCR. So I know that I just went through that very quickly, but based on what I said, can anyone tell me how, in this case, because again, we're dealing with G proteins, they activate, or they're active when they're bound to GTP. We've gone over this process a couple of times in different contexts. In this case, what would be acting as a GEF, which is when you have your guanine nucleotide exchange factor. Well, first of all, who can tell me what a GEF does? Opposite. Yeah. So it helps get rid of the GDP in the binding site for the G alpha, and instead GTP comes in. So based on what we know here, it's not a GEF, but what is essentially acting as a GEF in this situation? Yeah, exactly. So the GPCR, once it has the conformational shift when your signal binds, it causes a change in G alpha, which causes GDP to leave and GTP can enter. So in that sense, it's acting as a GEF. Does that make sense to everyone? Okay, so now the other side of that would be what is acting as a gap in this case? Who can tell me what a gap does? So a gap is not an enzyme. It doesn't have any catalytic activity. Yeah. Yeah, that's important. Yeah.
So if you're thinking about like back in, if you're looking at RAS, the gap in the protein's activating molecule, and so what happens is the catalytic like activity of RAS is hydrolyzing that GTP. So that protein comes in and speeds up the process and makes the process easier. It doesn't hydrolyze it itself. It just helps RAN or RAS or whatever G protein you're looking at do.
Yeah. Okay. Okay, what were you? Yeah, exactly. Yeah. So the RGS is able to bind to your G protein, activate its intrinsic catalytic activity. The RGS is not having any catalytic activity itself, but it's helping to activate your G protein, the G alpha, which is then hydrolyzing GTP to GDP, and that causes the entire thing to shut off. I think this might be a problem set question for you guys, or at least it's one that I've seen in the past, where then, you've inactivated this cascade, G alpha is now bound to GDP, it goes back, binds G beta gamma, then it goes back and forms the rest of the complex with the GPCR. If the signal is still bound to GPCR, what will happen?
as still be down to GDP, right?
So your G alpha is deactivated, it's bound to GDP. It forms that trimeric complex again with G beta gamma, but it goes back and the GPCR still has signal bound to it. So once it binds to the GPCR, what will happen to the G alpha? That's okay. So if the signal is bound to the GPCR, it's going to still be acting as a GEF. And once the inactive G alpha goes and binds again, it'll just have the same process occur again where the GDP leaves and GTP enters. So it's just kind of starting the whole process over again. Does that make sense? Yeah, it's just a continuous transduction of the signal, essentially. It's causing it to occur again. Because if signal, for whatever reason, is still bound to this GPCR and G alpha binds again, it's going to have the same function where it acts as a GEF and G alpha becomes active again, and then it can go on and transduce the signal through the rest of the cell. How do we feel about that? Okay. All right. So again, the GPCR is the GEF, the RGS is the gap. This is just another diagram that shows you the whole process where you have your signal molecule binding, the GPCR acts as a GEF, it causes the switch where GDP leaves, GTP enters, now your G alpha is activated. The activated G alpha no longer has affinity for G beta and gamma, so they dissociate from each other. The yeah, beta and gamma stay together. They continue as a complex, but G alpha no longer has affinity for them. GTP goes and activates whatever signaling pathway it's meant to activate. And then it will, at whatever point when this pathway needs to get shut down, some G alphas have like their own natural rate of catalysis is enough to hydrolyze the GTP to GDP, but others need an RGS. So you might have an RGS acting as a gap and causing that hydrolysis to occur. So then G alpha is now GDP bound, it's inactive, it goes, binds back with G beta gamma again, they bind with the GPCR, and then it starts over again. This is just same thing. So, some common misconceptions. It's a common one, so don't feel bad, but GAP and GEF are not enzymes. They just help activate enzymatic activity. They're effector molecules, activators, yeah.
Yeah, we had the import-export cycle on the first exam when I took it, and I was lost when I was driving to school without knowing that, and then I was playing back the review that Rosella and Amon had, and Rosella said that at the beginning of the report. That is one of the, that is a way that Laura is able to like ask a trickier question, I guess. And then for whatever reason, if you go on to think about how
yeah, like I don't think she would even ask that specifically. I think people just like they write it down as part of their answer.
Like, oh, and then gap goes in and hydrolyzes it.
Yeah, exactly.
So you would say gap speeds up the process.
Yeah, you could say, well, I think like the common blanket statement that Laura uses all the time is that GAP activates G protein's intrinsic catalytic activity. You can just say GAP activates the G protein.
That's what I say.
Yeah.
If you start your sentence off with, like, for the other pathway, like, oh, RAS is a GTPase. And then you say the gap is what's doing the intrinsic activity of like hydrolyzing, then you're basically saying that you don't really understand like what GTPase is.
Yeah, because if GAP were able to hydrolyze GTP to GDP, like it kind of,
You don't.
yeah, like the GTPase, it doesn't really have a role. But just remember, you know, like ACE at the end of a molecule usually means an enzyme, so that's the thing that's performing the enzymatic function. Okay, a lot of people just confuse which does which. So remember, I think that like knowing what GEF and GAP stand for is helpful because it tells you exactly what they do. Guanine nucleotide exchange factor, it's exchanging one molecule for another. It's exchanging GDP to GTP. And then GAP is the GTPase activating protein. Well, if you're activating a GTPase, what do? You hydrolyze GTP to GDP. So knowing what they stand for helps you remember what they're doing. Again, those are just the examples that we've seen before. And all of these pathways are highly regulated. So one thing that Laura likes to ask a lot is just different examples of, oh, you add in this regulator, what will it do? You inactivate this regulator, what will it do? And you just have to apply those concepts to what you know and just go step by step into how it would affect the pathway. So that could be activating a pathway, deactivating a pathway, all affecting the amount of signal that you have going through the cell. And, you know, for example, if you have an overactive MAPK pathway, that could lead to too much cell division, which could lead to a cancerous phenotype. You know, gain of function, loss of function, those types of things. Alrighty. There's some, thank you. Oh yeah, we'll accept applause, that's fine. Yeah, these are just some tips. You know, if you're able to look at a diagram and know what's happening without having to read and like be told what's going on in it, that probably means that you're understanding it pretty well. Laura always says, you know, try to teach these concepts to anybody. It doesn't have to be a person, doesn't have to be a living object, but, you know, explain it to something. Writing things down is better than typing. Making your study sheets is an excellent study method.
On paper without typing it.
Yeah,
I agree.
Laura used to not let people type their study sheets, and I think that was the best thing because that's how it's such a good study method. That's the whole point of why she gives you a cheat sheet, is so that you study by making the cheat sheet.
I also like so back when I was taking the class, I would meet up with like a couple friends at South College, and then there's like one specific room right by the hill that has like a moving whiteboard that they're kind of like tall, and then you just draw like a pathway hundreds of times, just like explain to each other and like do modifications and stuff like that. So I think the best way for you to be able to like write down that you know what you're talking about and you understand the pathway function, understand molecule function, understand the signaling, is for you to be able to explain to other people. Preferably people that have no idea what the pathways are, that you understand what happens. Because if you're explaining to someone, and this is important also for exam writing, I saw a lot of this while I was correcting. Let's try not to leave in information that we think is implied, because unless you write it, there's no way for us to grade it. We can't assume implied information on grading. And that, let's say that like you're talking about on the cell survival signaling pathway that like mTOR is going to be phosphorylating the AKT protein, right? If you don't say that there's a conformational change that allows for that to happen, you're not explaining how it happens. And so you're deducting that information that you think is like, so like, oh, this makes so much sense, right? The AKT is going to be phosphorylated by mTOR. You need to tell me why the mTOR is phosphorylating it, but not just why, also how it happens. And that I think is a good example. That's an example that like stuck with me about like how important it is for you to know how to explain it.
Also, I think the last bullet is the most important one because all of Laura's questions are application based. So, you know, I mean, you guys saw in exam 1, she's going to throw at you all these different hypothetical scenarios. And if you can explain how it's affecting these pathways, you know, like I said, if you add a phosphatase to the RAS RTK MAPK pathway, you know, how is that going to affect it? Like if you have a phosphatase targeting MAPK versus MAPK2 or whatever, you know, like being able to explain, well, okay, if I shut down this particular step of the pathway versus another step, how would a phosphatase affect differently in the survival signaling pathway? What would happen if, you know, the signal couldn't bind to an RTK? What would happen if the RTK couldn't dimerize, you know, and being able to really look at each step at which different processes happen and then insert this hypothetical scenario into where that would happen and how it affects everything else after that. And I think that's all we have.
Yeah. Yes, question. Sorry? It's gonna be posted on the TA YouTube channel, and we're gonna send it.
Yeah, there is. Like and subscribe.
It's recorded. Yeah, Gabe uploaded the recording. Like,
Uh, the link is on Moodle, I believe. I wish we're
Don't
not
think
paid for
so.
this. We do it out of the goodness of our hearts.
Well, the link gets sent out along with the
Oh,
presentation.
YouTube channel link for review sessions. Yeah. Well, it's there.
By
It has all
285
of them.
TAs. But the recording is gonna be uploaded, and then the uploading is gonna have a link being sent to your emails.
Yeah.
But yeah, so that's it. We're gonna stop the recording now. And then we are gonna stay here a little bit to